Hi guys, it's Jubilee again, and we're going to do another TA review. Hopefully you guys are seeing the light at the end of the tunnel around now. Good news, we only have 2 more quizzes and then your final. So we're getting close. You've already done your clinical correlations, hopefully. So yeah, just hopefully you guys are excited. We're getting towards the end. And these 2 quizzes only have 1 system each, so hopefully it'll be a little easier for you guys. But yeah, okay, so we're gonna go ahead and get started, and this week we're doing male repro. So the main functions of male repro, like many of the systems, it has exocrine and endocrine functions. And so the exocrine functions are mainly going to be, to produce sperm, right? To produce sperm and to make them viable so that they can hopefully impregnate a female, right? So they can meet up with an oocyte and create a zygote, right? And so not only do we need to make sperm, but we need to produce, maintain, and transport that sperm, and we need to make protective fluid to go along with it. And so when we talk about sperm, fully mature sperm that is fertile is going to be referred to as spermatozoa, okay? That's the technical term for it. And so while the sperm is being formed, we'll see that it goes through different stages, and so it's called different names, but fully mature sperm that is ready to go and is fully fertile and can create a viable pregnancy will be spermatozoa, okay? But yeah, so some of the other functions, we need to discharge the spermatozoa containing semen into the female reproductive tract during copulation, right? And then the male reproductive system also has some endocrine functions, and the main thing for that is just going to be producing the main sex hormones for males. So that's going to be testosterone, right? So this exocrine function is going to be going outside of the male body, and the endocrine is going to be staying within in the bloodstream. Yeah. So we're going to talk about the testes, and so that's the male gamete, the testes, and basically they're a bunch of tubes, okay? So the testes, being the male gamete, that's what's going to produce the fully mature spermatozoa. That's also where testosterone is going to come from. So our exocrine and endocrine, some of those secretions are going to come from the testes. And so it is a bunch of tubes while we're in all of these different stages of producing those fully mature spermatozoa. So the first tube that we're going to learn about, where everything starts out, is going to be what's called the seminiferous tubule. So all of these in here, these really curly windy things, are going to be seminiferous tubules. And that's going to contain spermatogonia. Okay, spermatogonia are going to be kind of like the origin cell that's going to make all of the germ cells. Okay, so it's going to give rise to the germ cells. And when we talk about germ cells, that's the all of those are going to give rise to spermatozoa. Okay, germ cells. So we'll see that there are different stages of those germ cells as they eventually develop towards spermatozoa. And then there's also going to be a third type of cell, the Sertoli cell, within those tubules, and those are kind of nurse cells, and we'll talk about that a little bit more. Okay, but that's all going on in the seminiferous tubules right here. So that's where we're making those early germ cells and developing them into early spermatids. Okay, and then once those cells are made, they're going to go through what's called the Reedy testes. Reedy testis, which is more just like a junction, right? They're all kind of draining into the Reedy testis. Then they're going to go through the efferent ducts, and we'll see in a second. You can tell the efferent duct is the efferent duct because it has true cilia. True cilia just means what we've normally learned as cilia, okay, on those endothelials, or on those epithelial cells. Okay, and it's also going to have a scalloped appearance to those epithelial cells. So that's how you can tell it's the efferent duct that we're at. and all those efferent ducts are going out, right? They're efferent ducts and they're going out, and they're going to go into this big long coil outside of the testis called the epididymis. Okay, so this whole thing right here is the epididymis. The epididymis, you can tell it's the epididymis because it has really long stereocilia, which are basically like fake cilia. They're not true cilia like we have in the efferent ducts. They're much longer. And this is where our final maturation as well as storage of sperm is going to occur. Okay, because we said we're making these germ cells, but we need them to develop into fully mature and fertile sperm. And so that's all going to happen in the epididymis. Okay, so if we took sperm from the seminiferous tubules and we tried to impregnate a female with it, it would not work. Okay, that sperm hasn't undergone all of the changes that it needs to go through in the epididymis to become fully fertile, motile, and working sperm. So that happens in the epididymis. So that's an important thing. And it will also store sperm towards the end. Okay, it'll store that sperm so that it's all there and fully mature at the end of the epididymis until it's ready to go. Okay, and once it's ready to go and we need that sperm to join the ejaculate, it goes through the ductus deferens. Okay, so the ductus deferens isn't normally holding sperm. It's only through ejaculation that we need the ductus deferens. And you'll see when you see the ductus deferens, it has a lot of smooth muscle because it's very strong, and it will really quickly expel the mature spermatozoa that we've made, and that happens through ejaculation. Okay, and when that happens, it's going to join with all of the other fluids that are being made in other organs of the male reproductive system, and that's going to form the full ejaculate. Okay, so ejaculate isn't entirely spermatozoa. It also has nutrients and buffers and just supportive fluids that are going to help the spermatozoa live through their journey through the female reproductive tract. Okay, so we talked about all those tubes, right? So now we're going to look at them. So the first one we said is going to be the seminiferous tubules. So these are the seminiferous tubules, and here's a zoomed out view so you can see it's all of these tubes in here. Those are all seminiferous tubules. They're all just cut different ways, but that was that really coily, curly stuff that we saw that makes up most of the testis. Okay, so these are the seminiferous tubules, and we said they're going to contain the spermatogonia, germ cells, and Sertoli cells. So we're going to go over that. So the spermatogonia, the way that you identify them is they're located on the base of the seminiferous tubule. So on the outside of the seminiferous tubule. So this is the outside lining of the seminiferous tubule. And the spermatogonia are going to kind of be anchored to the base. So you see how this one is flat up to the base? That's a really good example of a spermatogonia cell. Okay, and they're going to divide through mitosis, normal mitosis that all your other cells do, and produce a primary spermatocyte. Okay, so this cell is going to divide into two. One of those is going to stay on this base and still be a spermatogonia, and one is going to divide upwards and become a primary spermatocyte. Okay, once it's a primary spermatocyte, remember all of these are going to eventually turn into spermatozoa, so they're all germ cells. So once it's a primary spermatocyte, it's going to start to undergo meiosis. Remember, that's different. And that division is going to produce a secondary spermatocyte. Okay, so you can see right at this level, so all the cells are traveling from the spermatogonia at the base towards the middle of the tubule, right? So you can see once we go from the base, we have some primary spermatocytes coming off of the base, and then we have all of these mitotic figures happening. You see the condensing and weird looking nuclei, how they look kind of weird. Those are all mitotic figures, and so that's these primary spermatogonia undergoing meiosis down here. Okay, and so this spermatogonia is going to divide. It's going to make one new primary spermatocyte. That's going to divide through meiosis and create two secondary spermatocytes. And then those secondary spermatocytes are going to be 2N, right? Because that's how meiosis works. And they're going to undergo meiosis again to produce a 1N spermatid. Okay, so you can see how there's mitotic figures more towards the center. That might be a secondary spermatocyte undergoing meiosis as opposed to a primary undergoing meiosis, right? So they undergo meiosis, meiotic division twice, and that creates a spermatid, okay? Spermatids are not fully mature spermatozoa, but they are 1N, okay? They are that 1N that we need in our spermatozoa. And so once they become spermatids, these would all be early spermatids. They've just undergone that final division of meiosis. They're going to start to develop and become late spermatids. And so when we think of sperm, the whole reason we need sperm is to look the way that it's supposed to look is it needs to be a package delivery system, right? So once we have fully mature sperm, what we want it to be is we want it to be a very condensed package, and it needs to be motile and have a tail, right? So that's what these spermatids are trying to do. They're trying to get towards that. So you can see how down here we've got this big nucleus, and once we get more towards the middle, they're starting to condense. So these really dark black bullets are what we would call late spermatids because they're developing from this generic round looking spermatid that's still 1N to this more condensed late spermatid where we're starting to turn into mature spermatozoa, but we're not quite there yet. Okay, so that's going to be a late spermatid. So they're trying to look like these nice tight little packets in there. Okay, once they're late spermatids, they can go out into the lumen of the seminiferous tubules, and that's where they start to go through all the other tubules that we talked about, the Reedy testes, efferent ducts, epididymis, and during that process, they fully mature into our mature spermatozoa, okay? So that's all that's going on in the seminiferous tubules as far as our germ cells go. And remember we said we also have this third kind of cell called a Sertoli cell. And so what Sertoli cells are is they're basically nurse cells. Okay, so while these spermatogonia are dividing, spermatocytes are dividing, and everything, these Sertoli cells are there to support all of the germ cells within here. All of these are germ cells. And then another thing that they do is they're going to help create that blood-testis barrier. Okay, and we'll see some better examples that'll show you exactly what Sertoli cells look like and what they're doing. But I'd say maybe this is an example of a Sertoli cell, and you can see how it's kind of branching. Its cytoplasm is branching out around it. And what it'll do is connect to other Sertoli cells around this level within the seminiferous tubule. And that's to create that blood-testis barrier, because remember we said the nucleic material in these spermatocytes and spermatids is not the same as what the body is used to down here, right? We've got 1N and meiotic division undergoing here, and if our blood comes in contact with these cells, it will attack it, right? Because it recognizes these cells as a foreign invader. So we don't want that to happen. So these Sertoli cells kind of hold hands and create that blood-testis barrier as well as support the cells around them. Okay, and the reason they don't have to protect these spermatogonia is because, remember, those spermatogonia are just undergoing normal mitosis to make those primary spermatocytes. So until these primary spermatocytes undergo meiosis, we don't need to put them inside the blood-testis barrier. So that's why we've got our spermatogonia right here, and then our Sertoli cells will be right above that, making a blood-testis barrier. Okay, and then outside of the seminiferous tubules, we have Leydig cells. Okay, and we'll see them more in a second, but they're outside the seminiferous tubules. And remember, that's because the Leydig cells, what they do is produce testosterone. And remember, we said that's our endocrine function. So we want it to go into the blood, right? We want that testosterone to come in contact with the blood. So if we were to put it in these seminiferous tubules, put those Leydig cells and make them produce testosterone in here, that wouldn't be any good because we have this blood-testis barrier, right? So we put them out here because we need testosterone to help these cells develop. That's one of the functions of testosterone. We need a high testosterone environment in here to help these cells develop, but we also need it to go throughout the rest of the body and get into the bloodstream. So they are located outside of the seminiferous tubules. Hopefully that makes sense. Okay, so now we're going to look at some examples to help you guys identify things. So if we're looking at this toluidin blue stain, and we're trying to identify things, so this would be the outside of the seminiferous tubule, that outside lining. This would be that inside lumen of the seminiferous tubule. And if you look, this cell out here that's more towards the base, I would call a spermatogonia, okay? And then you can see right here, like this cell and this cell and this cell, you see how they kind of look different and how you can see their cytoplasm is branching out. Right? They don't look very circular like all the other cells do, very circular. These cells kind of branch out and connect to everything around them, and they're kind of anchoring down. Those are going to be your Sertoli cells, okay? Those are going to be your Sertoli cells, and what they're doing is holding hands at this level and creating that blood-testis barrier, okay? Once you get out here, you probably have a primary spermatocyte. A little later, you get some secondary spermatocytes. It's kind of hard to differentiate them because they look pretty much the same, but it's mainly just the location of where they are. Okay, primary are going to be lower than secondary. And then you'll have your spermatids really close to the center. So I'd say maybe this is a spermatid. This would be an early spermatid, and remember, as it starts to condense and look like a bullet, it's starting to become more like a mature sperm. It's not yet. These would be our late spermatids, okay? And once these late spermatids are ready, they'll go out into the lumen and continue on through the testes. Okay, so here we're going to look at an EM where you can see in a lot more detail. This is the outside of that seminiferous tubule. So down here at the base, we're going to have our spermatogonium. They're going to undergo mitosis, right? And so here you can really see, so this would be a Sertoli cell, this would be a Sertoli cell, here would be one, here would be one, here would be one. And you see how they're not very circular and they're kind of branching in between things and connecting. Does that make sense? How they have little branches going in between everywhere? Okay, so those would be your Sertoli cells. That's what's making that blood-testis barrier. And so after those, that's where you would have your primary spermatocytes starting to undergo meiosis, right? After the Sertoli cells, those nurse cells. And then you could also have some secondary spermatocytes undergoing meiosis. And then once that happens, you'll have your early spermatids and your late spermatids. So these would be your late spermatids, okay? But yeah, and so outside of that seminiferous tubule, remember we have blood outside here, not within here, because we have that blood-testis barrier. So we've got little capillaries, we've got some fibroblasts, and that's where we're going to have our lytic cells. Okay, so these are lytic cells, and you can see they've got some little secretions going on in their cytoplasm. These are our lytic cells outside of the seminiferous tubules so that they can produce that testosterone to help these cells develop as well as go into the bloodstream so it can affect the rest of the male's system. Okay, you've also got these little things called myoid cells on the outside of the seminiferous tubules, and that just kind of helps to contract down on this seminiferous tubule so we can keep the flow kind of going along within the lumen. Okay, so here are some more examples, and these really show you kind of what the Sertoli cells do, those nurse cells. So here you can see, this is kind of what I was talking about how they're not circular, like all of these germ cells are very circular, right? Instead, the cytoplasm kind of pinches and branches in between cells, and you see how this Sertoli cell and this Sertoli cell are kind of holding hands. That's that blood-testis barrier that they're making, so that blood cannot infiltrate through here and start attacking these cells that are undergoing meiosis, right? So that's what their Sertoli cells are doing. They're also just supporting all of these cells. They're kind of creating that supportive meshwork for these cells. So yeah, and here's an EM of that, so you can see this is a Sertoli cell branching in between two spermatogonia, probably. Oops. So there you can see how it's kind of branching and anchoring down in between those spermatogonia cells. Okay, here you can see more examples of seminiferous tubules and these lytic cells in between the seminiferous tubules, right, where all the blood and lymph vessels and everything are. So these are more examples of your lytic cells producing testosterone. Okay. So after we are done with the epididymis, remember, after we're done with the seminiferous tubules, so all of these are examples of seminiferous tubules. These are all seminiferous tubules that we just looked at, right, where we're creating those late spermatids. They're all going to meet and join into the Reedy testis. Okay, so that's that next kind of tube that they go into. So this kind of junction right here is the Reedy testis. Okay, and so that's basically what it is it's the junction of all these seminiferous tubules and what they're draining into. So this is the Reedy testis. This right here is starting to be the Reedy testis. Okay. Once it goes through the Reedy testis, it's going to go into those efferent ducts. And the way you can tell it's an efferent duct is because it has those true cilia, right? So just normal cilia. They're really tiny in light microscopy, right? Really tiny. These are those true cilia, and so that's how you know it's an efferent duct. And another thing is in humans, those efferent ducts have a scalloped appearance. And basically what that means is that some cells are taller than the others, right? So it kind of goes up and down and up, down, up and down. It just kind of pinches in at places because these cells aren't as tall as these cells. And so that scalloped appearance also really helps you know that it's the efferent duct. Okay. And then once we go through the efferent duct, so here's another example of the efferent duct. We're going to drain into that big epididymis, right? And so the way you identify the epididymis is it has stereocilia, okay, which is basically like fake cilia, okay? And so they're much longer than true cilia are, right? So this is an efferent duct. Remember, we've got that scalloped appearance of the epithelium and those really tiny little cilia right there. You can barely see them at this level, right? And here are the stereocilia of the epididymis, and you can see they're much longer, right? These are really long stereocilia all through in here. And then we've got pseudostratified columnar epithelium. And remember, we said this is the final location of maturation and storage of sperm. So they are not fully mature until they go through the epididymis, okay? So we get those spermatids in the seminiferous tubules, and they leave the seminiferous tubules. They go through the Reedy testes, efferent ducts, and the epididymis, and that's where they start transforming finally into mature spermatozoa, right? From spermatids to spermatozoa. So they just undergo all these changes and like fix their tail and everything like that, so that, and their acrosome and everything, so that they can be fully mature and fertile. Okay. A reason you have these long stereocilia is just to keep the sperm in the very middle of the lumen, right? We don't want the sperm infiltrating down in here, because remember we said that the blood can have a reaction to the sperm. So we're trying to keep all of these sperm in the middle of the lumen, and those long stereocilia help to do that. Okay, and then remember we said once that sperm is stored in the epididymis, it waits there until it's needed. And then the second that it's needed, it's going to go through this really big ductus deferens. Okay, this really big muscular ductus deferens. So this is part of that ductus deferens, and you can see all of this is smooth muscle around it, right? So it is a really muscular tube. And then we've got this epithelium lining it. So it's a really muscular tube. And the whole point of that is so that the second we want those spermatozoa to exit the epididymis, we can, and it's a really forceful motion, okay? It comes out really quickly so that they all come out at the same time. And so that's what the ductus deferens is there for, okay? So storage does not occur in the ductus deferens. It's more of a delivery system, Okay, this is a cross section of the spermatic cord, so you can see there are also a lot of really big veins as well as arteries in here in the spermatic cord, and then you can tell the one that's the ductus deferens because it just has so much smooth muscle, right? And then also it has a different epithelium lining it. Okay, so now we're going to talk about just the names of the processes that sperm undergoes. Okay, so we've kind of already talked about this, but so the whole process of the formation of sperm is spermatogenesis. Spermatogenesis. So we're making spermatozoa. Okay, so that's the name of the whole entire process, making mature sperm. And it's divided into some sub-processes that have very similar but different names. So don't let those mess you up. Okay, so the first one is spermatocytogenesis. Okay, and so that basically means we're making, Genesis is making, creating spermatocytes. Okay, so we're making spermatocytes. And the way, remember we said we make spermatocytes is going to be the division of the spermatogonia. So the spermatogonia are involved in spermatocytogenesis, and that's where they undergo mitosis. Remember, normal mitosis to create spermatocytes. Okay. Then those spermatocytes are going to undergo meiosis, right? So that primary spermatocyte is going to undergo meiosis. That secondary spermatocyte that's made from that is going to undergo meiosis again, and that's going to result in a one-end spermatid, right? So it's a one-end spermatid. It's done undergoing division, but it needs to become more mature, that spermatid. And so they undergo spermiogenesis. Okay, so that's that very last step. So that's the development of those spermatids into fully mature spermatozoa. Okay, so spermatids aren't completely done yet. So you can see here is like an early spermatid, right? And so it's got to fold in on itself. It's got to get rid of a lot of the stuff in its cytoplasm, rearrange its mitochondria, everything like that. It creates an acrosome, and that acrosome is going to help it penetrate and get into the oocyte for fertilization. So it develops that acrosomal cap on the very tip of it. It condenses its nucleus into that really tight bullet, and it starts to form this fully mature and motile tail so that it can move around. Okay, so there's a lot going on, and all of this is just spermiogenesis. Okay, all the other processes happen within those seminiferous tubules. Okay, and all of that together is called spermatogenesis. Okay, so if we're talking about spermatocytogenesis, that would occur near the base, right, where those spermatogonia are going to be dividing. Meiosis is going to happen more here in this middle. Spermiogenesis is going to start to happen right here, and it's going to finish and fully be done in the epididymis, like they said. Okay, so now we're going to do some practice questions. Which of the following cells is/are both correctly labeled and are not somatic cells? Okay, so they can't be somatic cells. And I guess I didn't explain this, but remember how we talked about all those cells that come from spermatogonia are going to be germ cells, right? Germ cells. So those are those new cells that we're making that are going to eventually become spermatozoa. And so kind of the different label for cells is they can either be germ cells or they can be somatic cells. And somatic cells just mean normal cells of the body, okay? Those are somatic cells. So we don't want a somatic cell. So A is a spermatid. So yes, this dark bullet looking thing is a spermatid. More specifically, it's a late spermatid, but yes, it's a spermatid, so that one does match. And it is a type of germ cell, so not a somatic cell, so that's good. So that one matches. B, Sertoli cell. And so this is a Sertoli cell. It's around at the level that Sertoli cells would be. It is a Sertoli cell, but remember, Sertoli cells do not divide and become spermatids and spermatozoa, so they are not germ cells. Those are normal somatic cells. Okay. And then this myoid cell out here, that one is also going to be a somatic cell, not a germ cell. Okay, so our answer is only going to be A. If I were to point to the secondary spermatocytes and stuff like that, those are also germ cells, and so that would work. But our Sertoli cells and our myoid cells are somatic cells, so that one doesn't work. Okay, next one. So gonocytes of the fetal testis give rise to which cell types labeled here? So gonocytes, it's basically just talking about the cells that come from the gonads. And so those are going to give rise to the cells that are going to produce the germ cells, right? And so remember when we talked about the cells that are going to undergo mitosis and become those germ cells, those are going to be our spermatogonia. And that's why they're called spermatogonia, and these are called gonocytes. Gonocytes give rise to spermatogonia. And so I already pointed to it. So yeah, that would be our gonocyte, more towards the base of things. This one's definitely undergoing meiosis because it's got those meiotic figures going on in its nucleus. So that one's not going to be a spermatogonia. That's probably a primary or secondary spermatocyte. B is a spermatid, so that's also not a spermatogonia. And so C would be our only answer. More towards the base would be our spermatogonia. And those come from gonocytes. Okay, this one. Of the three major events in spermatogenesis, which cells are involved in the named event? Okay, so these are those processes we were talking about. So A, it's pointing to these ones right here, and you can see they've got some meiotic figures going on in there, right? The nuclei look really weird and condensed within the cell. So that is meiosis, and that is what's going on right there during spermatogenesis. B, it's saying spermiogenesis. So remember, that's the formation of spermatids, from spermatids to fully mature spermatozoa. So here you can see this would be an early spermatid, and this is a really condensed bullet of a late spermatid, and that's a zoom in of right here. So it's very close to the inside. That's where we have our early and late spermatids kind of undergoing spermiogenesis. So that's correct. This one is mitosis. And so that's these cells right here. And so they do have those figures. So those could either be meiotic or mitotic figures going on in the nucleus. And the reason I know it's mitosis is because they are still under the blood-brain barrier of the Sertoli cells. Okay, so here is where the Sertoli cells are. I can kind of see those bluish granules from those Sertoli cells right around here. And so that's how I know it's still mitosis and not meiosis. So this is those spermatogonia undergoing mitosis to make those primary spermatocytes. So yes, that one does match. That one's all of the above. Okay, and that's it. Hopefully that was helpful. And good luck with your quiz, you guys.